In this presentation, we are going to examine allosteric enzymes. Those are enzymes that do not obey Michaelis-Menten kinetics. So, enzymes that obey Michaelis-Menten kinetics are not regulated. So what that means is that if substrate is present, the reaction occurs. And so curve is simple parabolic curve. Initially, reaction rate increases very high with increase in concentration of substrates until it reaches Vmax when enzyme is fully saturated. Which means if a substrate is present, enzyme will catalyze reaction without any regulation. And the more substrate is present, the faster the reaction until the enzyme is saturated when reaction rate plateaus. That's kind of expected. Now, the activity could be altered by regulatory molecules that reversibly bind to sites other than catalytic sites. And typical regulated enzymes are allosteric enzymes. And they are involved in regulated metabolic pathways. So allosteric enzymes are essential for proper function of regulated metabolic pathways. They do not obey Michaelis-Menten kinetics, and typical plot that shows activity, reaction rate of an allosteric enzyme as a function of concentration of substrate is sigmoidal plot. So initially reaction rate is very low until a certain concentration of substrate is reached, when reaction rate sharply increases until eventually it again reaches plateau or saturation. This is consequence of cooperative binding, and this is concept we are already familiar with because we have encountered similar behavior when we consider transport proteins, myoglobin and hemoglobin. And here is an example of regulation of allosteric enzyme by products, which is one of the principal ways how allosteric enzymes are regulated. So if we consider this metabolic pathway, hypothetic metabolic pathway, where starting material is material A, substance or molecule A. It is valuable starting material and it should be conserved unless it is needed. B, C, D, and E are intermediates in production of F, but B, C, D, and E have no biological roles, so they have no value. Their only value is that we have to convert A to B, B to C, and so on to produce F. And F is the final product. And it is needed in limited amounts and cannot be stored. An example would be ATP. F could be, for example, a molecule of ATP. So now in this sequence, we can see that each step of the sequence, it is a 5-step sequence, is catalyzed by different enzyme. So first step, conversion of A to B is catalyzed by enzyme E1, B to CE2, and so on. The first step commits conversion of molecule A into F, because A could be converted into a number of different metabolites. Action of enzyme E1 on molecule A commits that molecule to being converted to F, commits it to this particular, to the entire sequence where it's going to be converted to molecule F. So this step is called committed step, and enzyme E1 catalyzes the committed step. So for that reason, enzyme E1 should be regulated. There is no point in, let's say, regulating enzyme E3, because if enzyme E3 is regulated, that's conversion of C to D. And let's say there is enough F, we don't need any more F. So enzyme E3 is regulated, and then it stops through a regulation, through being regulated, it's deactivated, so it stops catalyzing conversion of C to D. Well, then first and second step would still occur and C would begin to accumulate, but it has no biological role. So very important, very valuable molecule A would have been converted to C, C would start to accumulate, and that would be actually very wasteful. So for that reason, enzyme that catalyzes committed step should be an allosteric enzyme, and is an allosteric enzyme, and that's one that is regulated. while enzymes E2 through E5 are Michaelis-Menten enzymes, simple enzymes that are not regulated and that simply catalyze reaction that if substrate is present, its reaction occurs. So if B is present, it's going to be converted to C. If C is present, it will be converted to D. If D is present, it's converted to E, and finally E to F. No regulation. But if A is present, enzyme E1 is regulated, and reaction may or may not occur. If F is needed, then reaction will occur. But if F is not needed, then how is enzyme regulated? Well, it's regulated through feedback inhibition. And feedback inhibition means that when F is produced in significant or sufficient amount, then excess of F appears because it's no longer needed. And then that excess of F reacts with enzyme E1 to form FE1 complex, and that deactivates the enzyme. And so that prevents committed step of the sequence to occur, and null and A is no longer converted to F. So this entire sequence is now arrested. It doesn't occur. So feedback inhibition occurs by binding of product to a site that is different from the active site of the enzyme, because F is not substrate for the active site of the enzyme. It binds somewhere else to the enzyme and changes shape of the enzyme so that enzyme is now inactive. And because it binds to a place other than the active site, that's why the enzyme is called allosteric enzyme, because it's allosterically regulated. And allosteric comes from allos, other, and stereos, space or structure. In this hypothetical example, we have two cooperative pathways which converge to synthesize the final product K. So starting with molecules A and molecules G. There are two pathways, so A is converted into intermediate F in 5 steps, and then G in a separate pathway is converted into intermediate I in 2 steps, and then F and I are starting materials which are in 2 steps converted into K. As before, A and G are valuable compounds that can undergo number of different transformations to be converted into number of other different valuable products. And so these are only some of the possible transformations or one possible pathway. So A should be conserved unless it is needed for synthesis of other materials. All the intermediates, so B, C, D, E, F, H, I, J, they are all only intermediates and they have no value other than being intermediates for production of K. So they should not be accumulated. It would be wasteful or potentially even dangerous if they were to accumulate in the cell. And their only role is that they serve as intermediates to produce K. K is important product, but should be produced as needed and cannot be stored. That means that these two cooperative pathways for production of K should be highly regulated. Now we can identify two committed steps. Step catalyzed by enzyme E1 is committed step for the first top pathway that involves 5 steps for production of F. So that's going to be an allosteric enzyme that is regulated, while rest of the enzymes will be Michaelis-Menten enzymes and will simply operate following Michaelis-Menten kinetics. So if starting material is present, enzyme will work. Enzyme will catalyze the reaction. And for the second pathway, bottom pathway, enzyme E10 catalyzes committed step. So that's the allosteric enzyme, while E11 will be Michaelis-Menten. And of course, once F and I are generated, enzymes E21 and E22 are also Michaelis-Menten enzymes. So regulation is going to be on the enzymes E1 and E10. But now inhibition of both E1 and E10 by either product F or intermediate F, product of each individual pathway F or I, would be inefficient. Because if F is produced in larger amount, for example, and it inhibits both E1 and E10, then that would cause F to accumulate. Because while F would inhibit E1 and prevent formation of any more F, if it were to also inhibit E10, it would inhibit formation of more intermediate I. And intermediate I is necessary to react with F to convert it to K. So that would result into accumulation of F. And same would be situation if I were to inhibit both E1 and E10. Then that would, the same situation would result in accumulation of intermediate I. So that would be very inefficient way to regulate these cooperative pathways. Much better is if F inhibits E1, so if F is present in excess, if it inhibits E1, so it prevents formation of more F, but it stimulates E10. That means it stimulates production of more I. So if F appears in excess, then production of more F is inhibited, or basically completely, or possibly completely stopped, while production of I is speeded up. Reaction rate is speeded up because enzyme E10 is activated and works faster than usual. So that would be a better way to regulate cooperative pathway like this. Besides allosteric control, there are other ways to regulate enzyme activity. Enzyme activity is regulated by temperature, but most of the reactions occur at physiological temperature, so this is not a very important way of regulating, in fact, if it's a way to regulate enzyme activity at all. But we can consider activity of enzymes or an enzyme as a function of temperature. And we can see that activity of an enzyme increases with increasing temperature up until certain maximum activity is reached. And then there is very sharp decrease past that temperature due to denaturation of the enzyme, due to unfolding of a chain, and enzyme becomes inactive. Another way to regulate enzyme activity is pH. So typically enzyme has a maximum activity at particular optimal pH, and it displays a bell-shaped curve. So it's at particular physiological pH where enzyme is designed to operate. Different tissues, different cells may have different pH. Optimal pH then would be characteristic of particular enzyme. And dependence of pH is consequence of presence of various ionizable groups at active site of the enzyme. So here is a hypothetical example where active site would contain glutamic or glutamate residue and arginine residue. So arginine residue could be deprotonated, glutamate protonated depending on pH, and that would affect activity of the active site. One of the most common ways to regulate activity of an enzyme is by inhibition. Besides regulation of activity of an enzyme, many drugs and many toxic agents act by inhibiting enzymes. Also, we may carry out research into activity of enzymes, and we can elucidate mechanism of action of enzyme by studying their inhibition. There are two types of inhibitors in general, reversible and irreversible. In case of reversible inhibition, typically inhibitor is bound to the enzyme through intermolecular interactions. And there are different types of reversible inhibition. It could be competitive, uncompetitive, non-competitive, or mixed uncompetitive and non-competitive. Irreversible inhibition involves inhibitor binding tightly through covalent bond to the enzyme. And types of irreversible inhibitors are group-specific reagents, affinity labels or substrate analogs, suicide inhibitors, and transition state analogs. Competitive inhibitor has a structure that is similar to that of the structure of the substrate. And that way, competitive inhibitor competes with the substrate for binding to the active site. Now, substrate and inhibitor have similar structures, so inhibitor fits nicely into the active site but doesn't cause any reaction. And in fact, its bonding to the active site is stronger compared to bonding of the substrate to the active site. So that inactivates the catalyst because inhibitor remains bonded to the active site and prevents any substrate from accessing it, and that stops the reaction or inactivates the enzyme. Some medications are competitive inhibitors, and an example is antibiotic sulfonylamide. So para-aminobenzoic acid is starting material, naturally occurring compound that bacteria need to synthesize folate. Inhibitor is sulfonylamide because it binds to the active site and deactivates enzyme that catalyzes synthesis of folate. Since in this case, in general, in competitive inhibition, substrate and inhibitor compete for the active site and binding is reversible, competitive inhibition can be overcome by adding more substrate. Another type of inhibition is so-called uncompetitive inhibition. An uncompetitive inhibitor binds only to the enzyme substrate complex. So once enzyme substrate complex has formed, an uncompetitive inhibitor has very high affinity for it and will bind to it. So uncompetitive inhibitor here is shown in blue. So enzyme would be that large white molecule, globular molecule, substrate green, and then inhibitor would be in blue. So it stabilizes enzyme substrate complex, which is usually unstable. But now that this complex is stabilized, it is stable, it simply stays as is and reaction doesn't proceed anymore, and both enzyme and substrate are taken out of the reaction. So that prevents any further reaction. Of course, this type of inhibition cannot be overcome by adding more substrate. But since binding of the inhibitor to the enzyme is reversible, then inhibition could be overcome by removing the inhibitor. Then next type of inhibition is non-competitive inhibition. A non-competitive inhibitor binds to the site that is different from the active site. So both non-competitive inhibitor and substrate can bind to the enzyme, but non-competitive inhibitor changes the conformation, the shape of the enzyme, so enzyme is no longer capable of catalyzing the reaction. So even if substrate is bound to the enzyme, reaction will not occur because active site can no longer catalyze the reaction. So non-competitive inhibitor decreases the turnover number of the enzyme. Just like uncompetitive inhibition, non-competitive inhibition cannot be overcome by adding more substrate, but can be overcome by removing the inhibitor. And then finally, we have mixed non-competitive inhibition. In a mixed non-competitive inhibition, a single inhibitor both hinders the binding of the substrate and decreases the turnover number of the enzyme. Now we are going to examine reaction kinetics of these types of inhibition. So competitive inhibition can be overcome by adding more substrate. The effect is that apparent value of Km, Michaelis constant, is increased because more substrate is needed to obtain the same reaction rate. Of course, maximum reaction rate Vmax is the same, but more substrate is needed to reach it. That's because substrate and inhibitor compete for the active site. So more substrate has to be added to overcome effect of the inhibitor or to outcompete inhibitor for the active site. In uncompetitive inhibition, the inhibitor binds only to the enzyme substrate complex, and then this complex is stable and doesn't give any product. Formation of enzyme substrate complex lowers the concentration of both enzyme and substrate that are available for the reaction. And so the result is lower value for Km and also lower value for Vmax. In uncompetitive inhibition, inhibitor binds either to the enzyme itself or the enzyme substrate complex. Because it binds to the site other than the active site, it doesn't matter whether enzyme is on its own or it has already formed a complex with substrate. The result is that value of Km is unchanged, but Vmax is decreased. The inhibitor only lowers the concentration of the enzyme, but not that of the substrate. And so the solution behaves as if it were diluted solution of the enzyme. And so this is all summarized in this table. And if you wish, you can pause the video here and examine the table and all this write-up in more detail. If we plot a double reciprocal plot of reaction with and without inhibitor, we can determine the type of inhibition that is involved. So in this first case, we have competitive inhibition. So the two lines, one that represents reaction without the inhibitor, that's the one shown in blue, and with inhibitor shown in red, have a common y-intercept. That's because y-intercept is 1 over Vmax, and in case of competitive inhibition, Vmax is the same. Of course, they have different slope. Slope is increased because in inhibited reaction, we have Km apparent, Km up labeled as Km up, and that's the apparent Michaelis constant. That's the constant of the inhibited reaction. That one is larger than the Km of uninhibited reaction. In case of uncompetitive inhibition, the intercept is increased because apparent Vmax is much smaller than Vmax, or smaller than Vmax. But slope is unchanged because ratio of Km to Vmax is the same in both inhibited and uninhibited reaction. So the two reactions give parallel lines. And finally, in case of non-competitive inhibition, the y-intercept is increased because Vmax apparent is smaller than Vmax. And the slope is increased because ratio of Km apparent over Vmax apparent is larger than Km over Vmax. But x-axis intercept is common. And that is summarized in this table. Again, you can pause the video here and examine this in more detail. Now we will examine irreversible inhibitors. Those are inhibitors that bind strongly through covalent bonds to the enzyme and dissociate either very slowly or not at all. They are sometimes used in research to determine the nature of or the structure of the active site. We can identify 4 different categories of irreversible inhibitors. They are group-specific reagents, affinity labels that are also called substrate analogs, suicide inhibitors, and transition state analogs. So, first group are group-specific reagents. Group-specific reagents react with side chains of amino acids, and so they inactivate the active site, but they also may bind elsewhere and simply cause the enzyme to change shape and inactivate it that way. So they will bind to any site that side chain that contains suitable functional group. Not just side chain that is present in the active site, but anywhere on the chain, on the enzyme chain that they can access. An example is iodoacetamide. Iodoacetamide reacts with thiol group with SH functionality, also called Mercapto group, of cysteine. And so it will bind to any cysteine residue anywhere on the chain. Whether that cysteine residue is on the active site or not, that doesn't matter. Any cysteine residue is potential binding site for group-specific reagent. For that reason, even though the name contains this term specific, these reagents are actually the least specific of all of the irreversible inhibitors. All other classes of irreversible inhibitors are more specific than group-specific reagents. These ones are the least specific. Simply, they don't discriminate between any sites of the enzyme or any side chains. Another class are affinity labels. Affinity labels are covalently bound to the active site residues and are structurally similar to the substrate. So they resemble the substrate, and that way they can enter the active site, but once they enter the active site, unlike substrate, which is bound to the active site through intermolecular interactions, affinity labels form a covalent bond. And now that's a stable bond, and enzyme is deactivated that way. Suicide inhibitors are modified substrates. So inhibitor binds to the enzyme, and then enzyme performs a catalytic reaction on it. As a result of action of the enzyme, then reactive intermediate is generated. And then that reactive intermediate inactivates enzyme through covalent bonding. This is more specific way than to use affinity labels. So affinity labels are more specific than group-specific reagents, and suicide inhibitors are more specific than affinity labels. Finally, transition state analogs are stable molecules that structurally resemble transition state of the enzyme catalyzed reaction. So, as you may recall, enzyme has the highest affinity for the transition state. That means that it will have very high affinity for transition state analog and will form very strong interactions with transition state analog. An enzyme has much higher affinity for the transition state compared to affinity for the substrate or, for that matter, the product. And that means that transition state analog will form much stronger bond to the enzyme than substrate would, so it's going to displace substrate and cannot be displaced by the substrate. Transition state analogs are highly specific, and they are the most specific of all of the types of irreversible inhibitors. An example of such inhibitor is inhibition of proline racemase, which is an enzyme that catalyzes isomerization of L-proline into D-proline. So it racemizes proline. It converts one enantiomer of proline, L enantiomer, into D enantiomer. And it's inhibited by pyrrole 2-carboxylic acid because pyrrole 2-carboxylic acid resembles transition state for this reaction. Finally, we are going to examine how metabolic processes are regulated. And there are three general ways in which metabolic processes are regulated. First one is by control of the amounts of enzyme, and that's genetic control through enzyme induction or enzyme synthesis. The amount of enzyme depends on its rate of synthesis and rate of degradation. So the level of the enzyme is usually regulated by change in the rate of transcription of the gene that encodes it, and that means the rate of synthesis, how fast it is synthesized. And so enzymes are synthesized or their synthesis is terminated in response to changes in the environment. This is a very slow way to regulate the amount of the enzyme, and it may take hours and sometimes days. Much faster way is to regulate catalytic activity of an enzyme. And catalytic activity of an enzyme can be regulated by, for example, reversible allosteric control, for example, by feedback inhibition. That one is almost instantaneous. It takes much less than a second, very short amount of time. Then reversible covalent modification. Examples would be phosphorylation of enzymes such as serine or threonine, those that have hydroxyl group, OH group in the residue, in their side chain. Regulation takes a bit longer compared to allosteric control and may take seconds. You will see that this is a very common way to regulate enzymes later when we cover metabolic pathways, actual metabolic pathways. Next, hormones may activate or deactivate enzyme, and regulation by hormones takes longer compared to covalent modification. It could take sometimes seconds, but usually minutes or longer. Some enzymes are also regulated by the energy status of the cell. An example would be adenosine triphosphate. High ATP level inhibits its further synthesis. And the third type of regulation is regulation of accessibility of substrates. So in eukaryotes, metabolism or regulation of metabolism is enhanced by compartmentalization. For example, fatty acid oxidation takes place in mitochondria, while their synthesis takes place in cytoplasm. Enzymes that are involved would catalyze either reaction, and so if both processes were to occur simultaneously, then we would have futile cycles that would keep going over and over. But compartmentalization segregates opposite reactions, and so the only one reaction can occur at a time. Also, another means of regulation would be control of accessibility of substrates to control the flux of substrates. So, for example, glucose breakdown can occur only if insulin is present to promote its entry into the cell. And this completes our lecture on allosteric control or general allosteric enzymes or control of the enzyme catalyzed reactions.